The exponential growth of bacteria occurs when there are optimal conditions inside of a culture container. We also see exponential growth of bacteria in natural conditions. So out in nature, we would expect them to grow exponentially. But when we get into the laboratory and we grow cultures over time, the conditions will not be optimal at all times. So we have to have an understanding of how bacteria grow in the laboratory for that reason. It turns out that bacteria have what we can describe as a growth cycle when they're being grown under laboratory conditions inside a container. They aren't going to grow exponentially the whole time that they're in culture. Instead, they're going to grow through these phases, again, that we call a growth cycle. There are 4 phases that we need to be familiar with. The first one is called the lag phase. The second one is the exponential phase, again, that optimal type of growth. The third is called the stationary phase. And the fourth one is called the death phase. Now, we can express this 4-step growth cycle graphically. You can see this when, again, you plot out cell number data over time. And that's what you're looking at on this slide. So on the x-axis, time is going by. All right, so this is the time that these cells are sitting in a culture container in the laboratory. And on the y-axis, once again, we have the number of cells. Now, we've gone again to the log paper in this example. So the exponential part of this growth cycle is going to look linear on this graph. So stage 1, we said, is the lag phase. Stage 2 is the exponential phase. Stage 3 is the stationary phase. And stage 4 is the death phase. Now let's think about what's going on in each one of those phases. The lag phase got its name because when you look at the growth cycle graph, the number of cells in the culture container isn't changing at all. Take a look at that again. From the time that the culture starts, from the time that we first inoculate the culture, for some period of time after that, the cell number is not going to increase at all. It's going to stay flat. That's the lag phase. Now, what's going on inside of the culture is that the cells have just started to go into binary fission. And remember, there's a lot that has to happen before the parent cell actually first divides into 2 daughters. We got to get through elongation. We got to build the divisome and build the septum and wait for the septum to contract and split the parent cells in 2. So yes, there is a period of time at the beginning of culture when you don't see an increase in cell number because the cells haven't quite hit that part of binary fission yet. So how long the lag phase is going to depend on the condition of the cells that you're using. How old are they? And also what the conditions of the culture are itself. In other words, what's the environment like in that culture container? What kind of media have you given those cells to grow in? Does it have all of the nutrients that they need? Is it at the right pH? And so on. A culture medium that doesn't have enough nutrients for the cells that you put in there is going to cause that lag time to increase. It's not that the cells aren't doing anything during the lag time. They are. They're metabolically active. They're making new macromolecules. Remember, they're going to be creating 2 new daughter cells pretty soon. So they're very metabolically active. They just haven't reached that first division stage yet. Then, of course, we enter the exponential phase. Each cell is actively dividing at this point. So you're going from one cell to two with each one of those parent cells. During the exponential phase of the growth cycle, the cells are really at their best. They are healthy, they are metabolically active, they are using what we call their central metabolism at this point. This is early on in the culture, so if everything's going well, there's plenty of nutrients in there. All of those cells are able to go through binary fission with no problem at all. They're going to use the kind of metabolic pathways that they prefer to use. So they're using, for example, glucose to make ATP if that's their primary metabolic pathway. If you were to pull some cells out of the culture during the exponential phase, they would be relatively uniform to each other in terms of how metabolically active they are. This is the best time during culture to pull cells out if you want to do some biochemical testing of those cells, or if you want to measure their generation time, or perhaps you want to harvest some of the metabolites that they're making through metabolism. Remember, we often grow bacteria for industrial purposes or pharmaceutical purposes because we want to collect some products that they're producing. And the exponential phase is a good time to do that. Now, eventually, the cells in that culture are going to hit what's called the stationary phase. And if you recall, the stationary phase is a plateau. Again, we're at a point where the cell number isn't going up or down. It's just staying nice and flat. So what's happening during this phase? Well, this is a time when the rate of cell division is being matched by the rate of cell death. Those cells have been in culture long enough now that some of them are going to start dying. So for every 2 new cells that get made, 2 of the old cells are dying. And that's why the cell number plateaus. Now, what's causing these cells to begin to die in culture? Well, primarily, it's because the nutrient levels are dropping and the waste product levels are increasing. So the conditions in the culture are not optimal anymore for growth. A lot of times bacteria will start going through what are called their secondary metabolic pathways. So if they're able to use alternative metabolic pathways, they will at this point. And that means they're going to start producing what are called secondary metabolites. For example, there are bacteria that are capable of making antibiotics, just like there are fungi that are capable of making antibiotics. And it's during the stationary phase that they often start to do this. The other thing that you'll start to see being produced during the stationary phase is spores, endospores. This is a time that's a little bit stressful, right? Nutrients are starting to run out. Waste products are starting to build up. This is a stressful time for the cells in that culture. So if they're able to sporulate, and remember for the pathogens, that's Bacillus and Clostridium. If they're able to sporulate, they'll start to sporulate here. So if I'm growing up, say, a Bacillus culture because I want to collect spores, I can start collecting them during the stationary phase. Ultimately, the cells are going to leave the stationary phase and enter the death phase. And during the death phase, the cell number will drop. If you recall, the death phase, which is the last one, shows a decline in the cell number over time. So what's happening? Well, cell death is occurring at a rate that is faster than new cells are being produced. And what typically happens is those cells, as they die, they're being lysed or degraded. So again, the number of cells in the culture is going to go down as these dead cells fall apart, basically. This is actually also an exponential process. It's an exponential loss here instead of an exponential gain. Now, this is another time to gather endospores if you're trying to harvest those from this culture. So you can gather endospores during the stationary phase and during the death phase of a culture. Eventually, of course, all of the cells in that culture container will be dead. You cannot maintain that culture indefinitely on a limited number of nutrients. If you look at some of the large commercial, industrial scale microbial cultures that are used to harvest the things that bacteria produce, what they do is they try to maintain their cultures in the exponential phase. They try to hold the culture in this particular phase. And the way they do that is through these gigantic containers that are full of culture media that constantly have old culture media drawn off and new fresh culture media added in. They're really amazing, these giant vats that are used to grow microbes. They have it down to a science in terms of getting rid of waste products out of that big culture vat and making sure that they're continually adding in new nutrients so that the cells will continue to divide in an optimal way.